Hi, my name's Tariq Gadar, and today we'll be talking about proto-oncogenes. So whereas tumor suppressor genes are, genes that sort of naturally in our body tamp down um, the growth and dividing of cells in our body. Proto-oncogenes are like a gas pedal, right? The tumor suppressor is the brake, the oncogene is the gas pedal, all right? And so this is the first of the pathways that I want to talk about, which is the PI3-AKT pathway. So let's work through this. It all begins when you get physiologically. So this happens normally in our body. Where cancer happens is where things get out of control. So that being said, where it all begins is when a growth factor, a cytokine, a hormone, we talked about these in the previous lectures, so go back and view those if you don't really know what's going on. They're going to bind to the receptor tyrosine kinase. That causes a change in the conformation of the receptor tyrosine kinase so that it cross-phosphorylates and recruits PI3K. All right? Phosphatidylinositol 3 kinase. All right? That's the function of PI3. That is the recruitment of PI3K. It happens when, and this is important, this is very important what I'm emphasizing because it underpins the whole idea behind how cancer happens. Normally, you get a growth signal that recruits PI3K, and then PI3K is phosphorylated and activated, and then it will transform PIP2 into PIP3, into phosphatidylinositol trisphosphate. All right? That's why it's called phosphatidylinositol 3 kinase, because what it's going to do is that it's going to catalyze the conversion of PIP2 into phosphatidylinositol trisphosphate, all right, or PIP3. So in cancer, what can happen is that there will be a mutation such that PI3K is always switched on. Okay? That's what can happen in cancer. And that's called a gain of function mutation. A gain of function mutation in the PI3-AKT pathway. All right, it can happen here. Well, let's just keep going downstream and see what happens. All right, so growth factors, normally growth factor, cytokine, a hormone binds, and then we get PI3K. PI3K catalyzes the formation of PIP3. And notice that P10 is a brake on this reaction. So PI3K is a gas pedal, P10 is a brake. Therefore, you would not be surprised if I told you that P10 is a tumor suppressor that is mutated in some kinds of cancer. All right, P10 is a tumor suppressor gene that can be mutated in some kinds of cancer because P10 is the brake on this reaction, whereas PI3K is the gas pedal in the reaction. So PI3K is a proto-oncogene that can become an oncogene if mutated, and P10 is a tumor suppressor gene. All right, and if mutated, it loses its function and it no longer suppresses tumor formation. Okay, what's downstream of this? PIP3 is going to lead to the activation of AKT. And then AKT through a lot of different pathways. I mean, it's going to activate mTOR, or the mammalian target of rapamycin, mTOR. It's going to upregulate NF-kappa B, and by upgrading both of those, you're going to increase cell growth, protein synthesis. You're just boosting metabolism, boosting growth, helping with division. That's what mTOR and NF-kappa B are going to contribute. And you can also lead to the inhibition of P53. And we talked about P53 in the last lecture on tumor suppressors. This is the guardian of the genome. All right, so the cell will not repair damage properly, and it will enter cell cycle even when it's not supposed to. That's what AKT helps promote. All right, now don't worry too much about how it does that. Also, it's going to lead to the prevention of apoptosis. So, okay, by activating mTOR, upregulating NF-kappa B, downregulating P53, and inhibiting apoptosis, you ultimately lead to cancer cell resistance, cell cycle progression, and cell proliferation. All right, and this can be upregulated in two different ways. You can get a gain of function mutation in AKT. You can get a gain of function mutation in PI3K. All right, and additionally, let's say you're in why do we see there's a link between inflammation and cancer? What if you're constantly getting inflammation in your body and you have this constant elevation of cytokines in your body? Then this pathway is more often than not switched on. right? So the effect of one of the effects of chronic inflammation is cancer, and this is one of the pathways by which it does it, because those inflammatory cytokines that are in the milieu, you might see this word again, so I'll spell it out for you, or milieu. Everyone pronounces it differently, but this represents the environment that the cell is in. So the cell's environment has cytokines and growth factors and hormones and all this stuff from inflammation that can lead to cancer. All right, and it can lead to cancer because what happens, you get activation of the receptor tyrosine kinase, recruitment of and phosphorylation of PI3K, activation of this reaction to form PIP3, the recruitment of AKT, the upregulation of mTOR and NF-kappa B as a result of that, which will increase cell growth and synthesis and metabolism functions. It's going to lead to inhibition of P53, which is going to increase cell survival and prevent repair of damage to the DNA. So normally you're supposed to repair DNA before you go into the cell cycle. That doesn't happen anymore. And lastly, it's going to decrease apoptosis. So when you put all those things together, you get in tumors that already exist, you will also get resistance to treatment, right? Because there's just another thing now that you need to target. So the current treatment might not work anymore. Additionally, you can get cell cycle progression and cell proliferation. And those last two are one of the hallmarks of cancer, right? Cell cycle progression that's out of control and uncontrolled cell proliferation are hallmarks of cancer. And that brings us to Mick. So Mick has a lot of functions. Remember, the overall theme here is I'm not trying to make you memorize random minutia. I want you to see the big picture. So broadly speaking, when you think of what Mick does, just think everything that a cancer cell needs. It's going to have, you're going to have more transcription. Here's E2F. Remember, retinoblastoma usually binds E2F and doesn't let it move to the nucleus, and it's the cyclin-dependent kinases and the cyclins in the cell cycle that sort of phosphorylate retinoblastoma, the RB protein, so that it comes off of E2F. So E2F can then be a transcription factor. Mick sort of enhances E2F. Let's just keep it simple and put it that way. All right. And as a result of that, it's kind of similar to when you lose function of retinoblastoma. So I kind of want to highlight this theme with the AKT pathway was, oh, PI3K is a gas pedal and P10 is a brake. Here, one aspect of Mick's function is that, oh, well, if retinoblastoma was the brake earlier, now Mick is the gas pedal, right? PI3K was a gas pedal, P10 was a brake. Here, Mick is the gas pedal, retinoblastoma is a brake. Okay. Also, it's going to increase protein biosynthesis, cell adhesion, all these other things that are required to development of cancer, et cetera, et cetera, et cetera. You can see here that there's a lot. We talked about the Wnt signaling pathway in a previous lecture, so we're not going to go into that here. But here's what I want to highlight. You're going to increase these and these. All right, you're going to have more cyclins, or more function of cyclins and cyclin-dependent kinases. You're going to have more E2F function. You're increasing the function of these two components, and that directly leads to progression in the cell cycle. So you want to ask yourself, what do you think? So I have a question for you. What do you think is going to happen if you get a gain of function mutation in Mick? You can develop one of the hallmarks of cancer. In this case, and these are the anaerobic metabolism genes. In this case, Mick does a lot because it's increasing transcription of genes related to growth. It's increasing the expression of and the translation of proteins required for the cell cycle, cyclins and the cyclin-dependent kinases. It upregulates anaerobic metabolism, and that's one of the hallmarks of cancer, acquiring anaerobic metabolism. It's called the Wallenberg effect, I believe, or Weisenberg effect. I'm blanking on it right now. But you don't need to memorize that. What you need to know is that there is a shift to anaerobic metabolism in cancer cells. And then some other functions that I don't want to harp too much on. But these are the three ones I really want you to keep in mind because they're also hallmarks of cancer. And that brings us to Burkitt's lymphoma. I have seen this tested a lot. I don't want to get myself in legal trouble, so I can't say where. I have seen this tested a lot, and let's leave it at that. And this translocation is pimpable on rounds. It's called Burkitt's lymphoma. All right, and what happens is normally c-Myc is under control of a regulated promoter, so it only switches on when events occur upstream of that to switch it on. When the body thinks there's a need for growth and division, etc., and repair, then there will be a signal transduction pathway that culminates in the expression of Myc. And that's on chromosome 8, and IgH, which is a part of the antibody heavy chains. That's what H stands for, antibody heavy chain. Okay, that is going to be on chromosome 14. Now you can imagine this Myc needs to be switched on and on depending on the context. But in an immune cell whose job it is to make antibodies, IgH is always going to be expressed, right? That's one of those more active promoters. Okay, so there's the concept of the weak promoter and the strong promoter. A weak promoter doesn't really bind transcription initiation as effectively, so the expression isn't too intense. A strong promoter, like the promoter for the IgH on chromosome 14 in immune cells, there's going to be a high drive. There's a need for it, and the promoter is a strong promoter, so there's higher levels of expression, relatively speaking, between IgH and c-Myc in any given immune cell. But what happens is that in a translocation, a piece of this chromosome comes off and attaches to this one, and it's a reciprocal translocation. So it's an 8-14 translocation. So this part of chromosome 8 that contains c-Myc comes off and attaches where? Directly downstream of IgH. What's the consequence of that? Didn't we say that IgH is a strong promoter? Right. Is c-Myc in front of a strong promoter or a weak promoter now? If it's next to IgH, it's in front of a strong promoter. So do you expect higher or lower expression of c-Myc? Higher. So what kind of cancer develops? Burkitt's lymphoma, a lymphoma, which is a growth of lymphocytic T cells of the immune system. It's a tumor of the immune system, of the not the T cells, of the lymphocytes. And we're not going to go into which lymphocytes right now, but those lymphocytic cells have a strong IgH promoter. So when you get a translocation of chromosome 8 onto chromosome 14, c-Myc is now under the influence of that strong promoter. All right, I'm going to say this is high yield. I have seen this tested twice, and I have been explicitly asked about what this translocation is on rounds. And then we have the Philadelphia chromosome, which is a similar story where you have a translocation. This time it's sort of a 922. All right, and this is the BCR-ABL. All right, and there's an increased risk of chronic myelogenous leukemia, CML. And that's really all I want you to know. It's a similar concept, except the fusion protein, this is kind of different from c-Myc in the sense that this isn't like ABL being put onto a strong promoter that is BCR. This is kind of different. This is a whole new protein. It's a chimera, a fusion protein. So BCR-ABL now has a new function altogether. Okay, because normally ABL is separate of BCR. Right, ABL is separate of BCR, but it comes together and it's gained function. It's a fusion protein. So that's what I'm trying to introduce here. There's two different ways, well, there's more than two, but I've introduced two different ways that translocations can cause cancer. In c-Myc's case, in Burkitt lymphoma, you moved c-Myc from its weak promoter onto a constitutively expressed or a strong promoter. So now c-Myc is constitutively, which means by default, constitutively by default expressed on a strong promoter. Here, ABL got pulled into BCR and now they have made a completely new protein, all right, with completely new function, all right? And I'm not going to go too much into detail. I just want to highlight a couple of things. Note here what's downstream of BCR-ABL. We have JAK signaling, right? We have our JAK-STAT signaling. We have our RAS signaling, and we know these are all growth pathways, right? And we see that there's some overlap on the downstream. If you go down far enough, we see that there's overlap with the AKT pathway, right? Okay, so in the grand scheme of things, and here's our good buddy Mick, but notice that it's not that BCR-ABL is elevating too many of these functions. This is a very complex protein map. What I'm trying to emphasize, I don't want you to get lost in the weeds of this diagram. What I'm trying to emphasize is notice how this new BCR-able protein, how it functions to upregulate multiple pathways, and those pathways also overlap with other ones that we've already studied. So what I'm trying to point out here is how there's different mutations that are all leading to the same result: growth, division, and survival, immortality. Okay, that is where all of this is heading. Okay. And you may have heard of HER2/neu. I just want to point out this mutation because it's testable. When we say triple negative breast cancer, we're saying ER negative, PR negative, so estrogen and progesterone negative, and then there's HER2 negative. That's the last part of triple negative. So HER2 is a receptor tyrosine kinase, and the reason why I'm bringing this up and that it's important is because you would use tyrosine kinase inhibitors to inhibit this pathway. Notice what's downstream of it: PI3K, AKT, mTOR. So we talked about mutations, and Mick is downstream of PI3K and AKT as well. And here you have RAS, RAF, ERK, MEK, ERK. So notice that you have the RAS growth pathway here, which you've talked about in a different lecture, so I'm not going to beat the dead horse here. And here we have the PI3K pathway. We talk about mutations in these. What you can also get is mutations in the receptor itself, such that the receptor is overexpressed, and that occurs in breast cancer and a few other types as well. But what we can do is we can target this aberrant protein using a receptor tyrosine kinase. And as an intellectual exercise, I want you to try to think, okay, how could a cancer cell resist a tyrosine kinase inhibitor or a TKI? How do they resist TKI? So a couple things can happen. First, it can change the shape of the receptor so that it no longer binds the drug well. Second of all, it could get a gain of function mutation down here, such that your tyrosine kinase inhibitor doesn't matter anymore. Because while the cancer may have started as the whole source of the, not the whole source of the problem, but the source of its hallmark of nonstop growth, it may have started with a nonstop switched on receptor, and you block that receptor and you made some progress. But remember, cancer cells are dividing so much, they're always accumulating new mutations. So what might have happened is just one of those cancer cells has a mutation, let's say in PI3K, a gain of function mutation in PI3K. Now it doesn't matter if you block the receptor tyrosine kinase, because this one cell that survives over all the other ones has a natural selection advantage, and it disseminates. And there's a couple of other things I just want to mention. You have the BCL2 1418 translocation in follicular and undifferentiated lymphoma. You have c-KIT upregulation in gastrointestinal stromal tumors. And these are both, this is an anti-apoptosis gene. When you think of BCL2, you want to think of anti-apoptosis. Increased function of BCL2 prevents apoptosis, because there's a protein called BAD and BAX and BAC, and the BAD family of proteins, they were named because they do bad things to cells. It causes apoptosis. BCL2 sort of traps BAD and prevents it from doing that. That might be an oversimplified way of explaining it, but it's easier to remember. So BCL2 prevents apoptosis. So you can see how this 1418 translocation can cause lymphoma. It's because you get, inhibition of apoptosis in these lymph nodes. Because remember, in the lymph node, you have lots of lymph cells that are proliferating, etc. c-KIT, you just want to think growth. RAS, we've talked about that. RAS is a growth pathway. RAS, RAF, MEK, ERK. And then you have RET, which is implicated in multiple endocrine neoplasia. And we've already sort of I'm not talking about their mechanism of action. You should have already a good picture of how proto-oncogenes work based off the examples we've already done. And that sort of completes the cancer genetics. I know we didn't really do cancer genetics, but sort of the proto-oncogene and tumor suppressor, this growth and promoting growth, preventing growth, that balance. I think we've talked about it at this point, and this will be the last lecture in that sort of genetic series.